Welcome back students to Yupp Master, and this is the botany class. We are going to conclude the plant kingdom today. This is the 7th class of plant kingdom. So far we have discussed about the general characters of the members in the Kingdom Plantae. We discussed separately about Thallophyta, means the algae, Bryophyta, Pteridophyta, Gymnosperms in the previous class. So the thing which is left is now Angiosperms, and the last topic is life cycle patterns or the alteration of generation. Okay, so whenever the question is going to be asked from the plant kingdom, it is most of the cases, as we are following the previous year questions, in that cases we have noticed this thing that the questions from the plant kingdom were comparatively much easier and were direct to the theory lines. Okay, but whenever I am using the word direct from the theory line, it doesn't mean they are going to copy paste the lines from the NCERT book. It may be not. So when they are going to make a typical question or a difficult question from this particular chapter, they may give certain things about the comparison among different members. Okay, so this is how we need to understand the theoretical subject. If you are going through any of the theory part, whatever is the subject, if it is theory, that means that there is a chance that you may get confused because of the language. So whenever you are revising the things, you are going to the same chapter again and again, it is necessary for each and every student that they must prepare a short brief note having the tables of the comparisons, making the flow chart so that the things can be more simplified. And this is how the thing goes. For example, if for flowering plants I am going to compare with the Pteridophyta, Bryophyta and Gymnosperms, I can easily find it out that who is the more closer one to flowering plants. Who is more closer to flowering plants? We can count like this. We can go in this way. The first is, like in flowering plants, the similarity with the Gymnosperms are more as compared to Turito and that too from Bryophyta. So like Gymnosperms, the Angiosperms are showing what? One, the seed formation. Number two, all are heterosporous. Number three, they are forming two type of sporophylls. So it's obvious that they will bear two type of spores, Microspore, Megaspore, which are going to form the male and female gametophytes separately. Dioecious gametophyte, that too what? Dependent. It is just like Gymnosperm. Like Gymnosperm, they are showing the event that is called as pollination, and this is how we say one more thing. During fertilization, we are observing the participation of pollen tube. That's why fertilization is siphonogamous. So these are affinities among flowering plants and Gymnosperms. Okay. Next, the Pteridophyta, only like Pteridophyte. How Angiosperms are resembling Pteridophyte? I am asking the question in this term. Or which characters of Pteridophyta are resembling to Angiosperms? The one, obviously these are land plants. It is not about their habitat. It is not about they all are land plants. It is about something else. Only to Pteridophyta. That like Pteridophyta, Angiosperms are vascular. The sporophytic plant body is dominant and independent, which is differentiated truly into root, stem and leaves. So the similarities are getting lesser. The similarities are much more less when I am going to compare with the Bryophyta. Only with Bryophyta, I am saying the Angiosperms are representing Bryophytes in terms of what? One, they form the embryo, means they are the members in the group that is named as Embryophyta. Nextly, I can say the fertilization in Angiosperms as well as in Bryophyta. Oogamous, internal sexual reproduction they are exhibiting, fine. So this is how we have compared. Now, if anyone is asking to you that out of all the land plants, Angiosperms are showing most similarities with Gymnosperm. Angiosperms are sharing most of the similar features with Gymnosperms. Okay, least similar features with Bryophyta. Okay, that type of question can be created. Now, this is also reflecting one thing, that Bryophyta were first, evolved into Pteridophyte, more evolved are Gymnosperm. No doubt Angiosperms are much more better in terms of their habit and the habitat in which they are found. So the flowering plant means the Anthophyta. See, the word antho means flower itself. Here antho means flower itself. So I can say anthology is a study of flower, not flowering plant, study of flower. Okay, anthesis means opening of flower. So such kind of things we can explain, right? So about the general characters of the group Angiosperms. One of the things that we can mention here as a unique character, 3 unique characters are there for Angiosperms. First is about their sporophylls, means the micro and megasporophylls are getting aggregated into flower. Now flower is going to be the place which is the place where pollen grains are formed, ovules are born. Spatial structure which are bearing the pollen grains and the ovules. Although pollen grains here representing the male gametophyte and ovules are megasporangia. The ovules are enclosed in the ovary. So as ovules are enclosed in the ovary, we can say the fruit is going to have the seeds, or I am more sure about one thing, that is ovules after fertilization forms the seed and ovary on fertilization forms the fruit. So the seeds in angiosperms are present in the fruit. The converse of the statement is not true universally. Fruit may have seed, may not have the seeds. Okay, but fruits, if they are considered in the cases of angiosperms, I can say the seeds are present in fruits. Fruits are protecting the seed inside them. Okay, most of the time. Fine. The seeds, if not present, the fruit is called as parthenocarp. For that detail, you can go for the classes of morphology of flowering plants. Fine. So these are the points that to be kept in the mind. Remember the unique feature number 1, and that is flower. So here are the characters that we need to discuss about. The one thing is that they are diverse, not only in the terms of taxonomy, they are diverse in the terms of habitats. Even the marine members are known. Zoospora, which we are going to study in the later chapters, like sexual reproduction in flowering plant. Zoospora is marine, clear, found in oceans. Okay, whereas many aquatic plants that are growing nearby your house, or you can say in the rivers, in the ponds and the lakes, you know many of them. Other thing is like Wolffia is the smallest. It is about the habit. They may be for few millimeters. Clear? Wolffia is almost microscopic type, whereas Eucalyptus is the tallest angiosperm or the largest angiosperm. I am not saying the tallest tree. It is tallest among angiosperms only. Some members are parasitic or saprophytic, and as well as we are saying they are divided into 2 classes. The 2 classes of angiosperms are dicotyledon and monocotyledon. Clear? Those will be dicotyledon and monocotyledon. This is how we are going to classify angiosperms into 2 classes. Let's see minor differences or just general differences, not too much in depth, just general differences. So far what we have studied previously in morphology, we can compare monocot and dicot on the basis of those morphological features as well. Like the seeds with 2 cotyledons in dicot, having 1 only cotyledon in monocot. We are saying the venation is reticulate. What is venation? The leaf is having this kind of venation, mid vein is bearing these veins, and these veins are further forming a network. This is reticulate, whereas in the cases of monocots, the venation is parallel. This is the leaf type, and we are saying that these are the veins. They are not getting crisscross, they are almost parallel. Okay. Nextly, the taproot system is common in dicots. So we are saying that the primary root that arises from radicle is bearing the lateral roots. But in the cases of monocots, the primary roots are short-lived, just like Pteridophyta. Okay, the primary roots are short-lived, and therefore the roots are getting replaced, and adventitious roots arises maybe at the base of the stem or some other parts of the stem. Okay, so this is different. The base of the stem bearing these type of roots, where the primary and the secondary roots are obviously not distinct. The secondary growth is common. Now, what does it mean by saying the secondary growth? Because it is not common in monocots. It is on the behalf of vascular bundles. Okay, this is on the behalf of vascular bundles. Now, Unique character number 2. Second unique character. I'll tell you here only. What is this second unique character of flowering plants? Now this is about vessels in xylem. Clear? We are saying vessels in the xylem is another characteristic feature of flowering plants. Okay. So now the xylem tissue is having two kind of conducting elements. Xylem conducting elements in angiosperms. Conduction by one is called trachees, the primitive one which is seen in Pteridophyta. Gymnosperms, most of the gymnosperms are having trachees only, and angiosperm is having, in addition to trachees, vessels. Okay. So this is why we are saying this is the second unique character of flowering plants. So the vascular bundle comprises of xylem and phloem tissue. This is one vascular bundle. Now this side I am saying this is xylem. Same on this side. This is xylem. Now the unique thing is existence of this structure here, which is named as cambium. Whereas this is P for phloem. Here also this is P for phloem. There is no cambium. Now cambium is meristematic tissue, means dividing cells form a group which is meristem, and cambium is one of the example of meristem. So we are saying here meristem is present within the vascular bundle and it continues to divide or it continues to grow. So open means nothing other than open growth. The cells are dividing always. That's why they are calling it as open. Whereas here it is written as closed, means cambium is absent. Okay. This is anatomical comparison between the monocot and dicot. This is going to be covered in the later classes that we used to study in anatomy of flowering plants. Okay, later we will discuss. So far we have discussed the two unique characters of angiosperms. One is about the sporophylls getting aggregated as flower. The second unique character of angiosperm is about presence of vessels in the xylem. Okay, it is not universal in angiosperms, but if vessels are present, they are most likely in angiosperms. They can be seen in exceptional cases of gymnosperms. Fine. Nextly, about the sporophylls. So the sporophylls are of two types. First type we are going to discuss is microsporophyll. What is microsporophyll in that case? The microsporophyll is represented as stamen. Stamen is the structure which is going to represent the microsporophyll. Now this microsporophyll is having two main parts. Two main parts I said. I am not saying only two parts, two main parts. One is filament, other one is anther. Out of this, the anther is the place where microsporogenesis will occur, or I can say that this is the structure which we are considering. Clear? This is anther. Okay. This one is filament. Now, in this way, the anther is having what? Lobes. These lobes are equated as or representing the microsporangia. Microsporangia is having several microspore mother cells. Ploidy obviously 2n. The ploidy of this structure is also 2n, anther also 2n. So microspore mother cell 2n is undergoing meiosis. Fine, meiosis will take place, means sporogenesis, microsporogenesis, and this is the structure which is getting formed here as microspores, also written as pollen grains. You can say the microspore is the first cell of male gametophyte. Okay. So this is how we are saying the anther is going to be significant in terms of forming the microspores. First type of spore is obtained now. The second thing, the male gametophyte is pollen grain, just now I told you, and this pollen grain is actually two cell stage, dispersed at two cell stage. I am saying dispersal. Whereas the mature pollen grain is having 3 cells. Okay, the mature pollen grain is having 3 cells. What are those 3 cells then? These 3 cells means, what are these 3 cells? So first I can say a vegetative cell, one only, okay, then a generative cell which will divide further. Clear? This generative cell will divide further. It will do mitosis and will form 2 male gametes. You can call them as microgamete as well. Ploidy as usual, because the structure is haploid in which they are getting formed by mitosis. So no change in ploidy, they remain N, haploid. Although the question is about other characters. What? The 2 male gametes, both are functional. Remember, in gymnosperm we have studied only one of the male gamete is functional. Number 2, out of these 2 male gametes, what we are discussing over here is that they both are non-motile also. Okay, these are the characters that you need to remember about the male gametophyte or the pollen grain in angiosperms. Male gametophyte in angiosperm is 3-celled, whereas what we have studied earlier means in gymnosperm they were either 5 or 6-celled, comparing in Cycas and Pinus, or they were 13-celled in Selaginella, and the gametophytes were multicellular in Bryophyta. So as we are moving from Bryophyta towards angiosperm, means the lower group to the highest one, we are observing that the size of gametophyte is getting reduced, or the sex organs are getting reduced and the number of gametes are getting lesser. Remember, as we are moving towards the advanced group, as we move towards advanced group of plants, one, sex organs, reduction in size, they are getting minimized. Number 2, gametes are getting lesser in number. That type of feature we can relate here. Okay, remember this is the side thing, means we need to remember that this is a logical type of question. Okay, considered to be that a student who has read the entire syllabus of the class 11th and 12th NCERT, so the examiner is thinking that, or supposed to be learned, supposed to be known to every of the student, those who are appearing for the NEET exam. So be clear about this. Okay, every time you are not going to get the lines just copied and pasted from NCERT book. Next line. The next thing, the Megasporophyll is actually carpel. The carpel is forming the Megasporophyll. Later on, this carpel, maybe one or many, are going to form the pistil. The pistil appears like this. 3 parts: stigma, style and ovary. What are the fate of these parts then? So I am going to tell about the each and every part. The stigma means receptive for pollen grain. Okay, fine. So the pollen grain are going to come over stigma, not directly to the ovule. Where are ovule in this? The style is what? A tube. Depending on the example, either hollow, solid or semi-solid, and providing the path to reach to the ovary. Ovary is the place which we are saying that bears ovule, and ovule is nothing other than what? Megasporangium. The ploidy is 2n. The ploidy of ovule is 2n, diploid. Okay, that kind of structure we used to mention. Remember all the things. How the ovules are distributed in the ovary, that we used to study as placentation. Okay, that is called as placentation. Next, so how does ovule appears or looks like. So this is an ovule. The diagram is taken from the 12th class NCERT because it is more relevant to study. We had to study all the things with respect to this diagram only. So here it goes that we have this most important structure which is called as embryo sac. In morphology, students, we have studied about hilum. Clear? What is hilum? Hilum is the place from where we are saying this is the ovule body, this much. Clear? This much is the ovule body which is getting attached to the stalk. This is the stalk. At which place? That is called as hilum. Okay, that is called as hilum then. So, see this hilum, place of attachment of ovule to the funicle, right? That kind of structure. Micropyle, just like earlier, means like gymnosperm is the apex of ovule, but here no pollen chamber is present. There is no pollen chamber on the micropyle. Okay, the pollen grains are first coming to the stigma, then reaches towards ovule. Okay, so here we are saying that this is the condition that we need to know: outer integument, inner integument, integument ploidy, 2N. This is nucellus, a vegetative tissue right now, parenchyma, simple tissue, which is going to differentiate to form megaspore mother cell. Clear? This nucellus, not nucleus, nucellus is going to differentiate as megaspore mother cell. Now that megaspore mother cell will result in megaspore mother cell whose ploidy is 2N. Okay. Now it will undergo meiosis and will form the megaspore. Now that megaspore is only the embryo sac. This megaspore is only going to develop as embryo sac. So here is the structure which is called as embryo sac now. Let's define the structure or characters about this structure. What is it? So what are these structures on the diagram right now? Chalazal and micropylar end. Although here chalazal pole is given on this side, micropylar is given on this side, means the diagram is just inverted. Okay, the diagram is just inverted. Be careful. Chalazal end means the base, micropylar end means the apex. Okay, or you can say this is the upper side and this is the lower side in actual. So count the number of cells on the chalazal side. The megaspore is now having what type of things? Remember, I can tell you again, this is the nucellus. What happened at the nucellus? As I was telling you, that nucellus is the place what is going to be there. Megaspore mother cell, only one, ploidy 2N, undergo meiosis, forms megaspore. Now megaspore will develop into embryo sac. The embryo sac whose diagram is mentioned on the this right side. So this is embryo sac. Now please mention these characters which are shown in the diagram. Clear? What are the important characters here? One by one. Towards the chalazal side there are 3 cells. How many? 3 cells. They are named as antipodals. How many in number? 3 cells. And 3 nuclei as well. Fine. In the center you are observing central cell. Central cell is this much big. Central cell means the biggest one. Largest cell of embryo sac. No doubt about this. Now count the number of nuclei here. How many? 2. These 2 reddish structure. These are 2 nuclei which are named as 2 polar nuclei, you can say. 2 polar nuclei. Okay, cell is 1, nuclei are 2. Cell is 1, nuclei are 2. Egg is only 1, then synergids. Now in total, 1 is egg, 2 are synergids on this side. Which side? Micropylar side. So here I am saying 3 cells. comma 3 nuclei forming egg apparatus. Okay, they are forming the egg apparatus. So when we are comparing 3 cells, 3 nuclei, 3 cells, 3 nuclei on this side, 2 polar nuclei, 1 cell, if I am going to calculate the numbers, then the numbers are like this. The numbers will be like this. This structure appear to be 7 celled. Count it please. Cells, 3 cells, 1 cell, 3 cell. How many in total? 7. Then count the number of nuclei. 3, then 2, then 3. Total 8. Seven celled, 8 nucleate. This is the typical embryo sac. Most of the embryo sac are like this. The most common type of embryo sac is this one. Most common type. Fine. Wait. Other questions. Antipodals are haploid. Each of cell in egg apparatus haploid. But if the largest cell or the central cell is put as a question, that what is the ploidy of central cell, this central cell is diploid, because sooner or later these two polar nuclei are going to fuse. They are going to fuse to form what? Definitive nucleus or secondary nucleus. So this is the example that we can mention over here, right? So this is how we are saying the arrangement. Then again see this, 7 celled, 8 nucleate is fine. Now how the nuclei are arranged? The nuclei are arranged in this manner. 3, then 2, then 3. So 3 plus 2 plus 3 arrangement. Clear? The arrangement of nuclei in the angiosperm embryo sac is 3 plus 2 plus 3. Okay. In the previous one, means in the gymnosperm, the megaspore was developing into the female gametophyte. True. Here also the female gametophyte. But the female gametophyte in the gymnosperm was endosperm. And here the female gametophyte is embryo sac. Clear? So that is the difference. Be clear about this. Fine. So here I am going to tell the feature, the comparison. Actually there is one line that you can write the entire information in one single line. How come? See this one. The megasporangia or the ovule differentiated to form one only megaspore mother cell whose ploidy is 2N, means diploid. Okay, whose ploidy is 2N. Next, meiosis. Functional megaspore. Ploidy, and after meiosis the ploidy will be obviously half. The number of chromosome will be half. The ploidy is going to change. So the functional megaspore in the two places. One is gymnosperms and the other is angiosperms. Okay, let's compare the two. In gymnosperm, this megaspore develops into female gametophyte Yeah, true. They are also female gametophyte. But the name of female gametophyte in the two groups are not same. Here in this example means in gymnosperm the female gametophyte is endosperm. Here the female gametophyte is embryo sac. So the two structures are not at all same. Development same. Clear? Development same. Here what I am going to say this, that particular line, that golden line is the endosperm in gymnosperms and the endosperm in gymnosperms and embryo sac in angiosperms are ontogenetically similar. Ontogenetically means development. This is not the word which we are studying for the first time. We studied this word ontogeny for the first time in living world. Clear? So ontogeny means development. Development is from the same structure. That's why they are ontogenetically same. Although the fate, the role of endosperm and this embryo sac is obviously different. This endosperm in gymnosperms, this endosperm in gymnosperm is first of all obviously haploid, but it is multicellular. Number two, the endosperm is nutritive. Remember hen's egg. Before fertilization, nutrition of embryo. Nutritive for embryo. Now before fertilization. Here embryo sac. Here embryo sac in angiosperm is 7-celled, means comparatively reduced structure. From multicellular it is becoming few celled in angiosperms. Number two is the site of embryo formation or embryogenesis. The fate is totally different. The two things are not exactly same. Angiosperms are also going to form endosperm, but that endosperm will be something else. It is not here. Okay. So remember how the two gametophytes are same or how they are different. Fine. Hope you are getting this. Next is fertilization in the group. Now fertilization is just like gymnosperm. It is internal oogamous chemotropic. Remember the pollen tube will grow because of chemical stimulus. Siphonogamous, the pollen tube will participate and the result is oospore. No doubt about this. But there is a difference in the cases of what? Pollination. Pollination is however different. In angiosperms. How? In gymnosperm we have studied the word that is called as anemophily. Number two, direct pollination. Here it is. One, the pollination is indirect. Why? As pollen grain landed on stigma. And number two, mostly entomophilous. Now what does it mean by saying entomophilous? So it is like when I am explaining the terms, it is like that indirect means pollen lands on or pollen grains. Stigma, not the ovule directly. Mostly entomophilous. Entomo represents insect. Majorly by insects as pollinating agent. So this is how the things are same or different. That's it. This is about angiosperms, fertilization, pollination. The two things are very clear now. Nextly, about the event which is third unique character of angiosperms. I repeat, third unique character of angiosperm is double fertilization. So another characteristic feature or unique feature number 1, 2, 3. Previous two were one, formation of flowers from the from the sporophylls, and the second is occurrence of vessels in the xylem tissue. Here is double fertilization. Now what double fertilization actually means? Fertilization is not that much simple. Actually here one more structure is going to participate, means the central cell or the secondary nucleus will participate. So there are two terms which are included in double fertilization. One is syngamy, other is triple fusion. Now how the two terms are different? Clear? This one, the two male gametes are functional. Both of the male gametes are functional. Now how they are going to participate? Here in the syngamy, the male gamete, here also male gamete. Ploidy of the male gamete is obviously N, is N. Okay. The other structure, female gamete or the egg cell, ploidy N. This side the structure is totally different one, and that is what? The two polar nuclei you can write, or even you can say the one secondary nucleus, whose ploidy will be therefore written as what? N? No, it will be 2N then. Okay. So what is their result? What is their result actually? If these are going to fuse, they will form zygote. Now that is called as amphimixis or fertilization. That resulted in what? This resulted in zygote, or you can call it as oospore. Whereas this structure means the male gamete when fuses with the two polar nuclei is called as triple fusion. Now I think you can understand this thing. One nuclei and two nuclei, three together. That's why triple fusion or one secondary nucleus. So here we are saying this is resulting in vegetative fertilization or trophomixis. You will come to know why it is named so. So this is resulting in formation of PEN, whose ploidy will be then N plus 2N, 3N. This PEN is primary endosperm nucleus. Okay, fine. Here endosperm is going to be the result of fertilization. Here endosperm is the result of fertilization, whereas in the cases of gymnosperm, the endosperm was formed prior. So here is the comparison. Gymnosperm, like aves, forming, or like the birds, like the hen, they are forming the nutrient, means the yolk of the embryo prior to fertilization. Whether fertilization happens or not, whether zygote is there or not, the nutrient will be available. And in the cases of angiosperms, we are observing these are something like mammals. First the fertilization occurs, which is going to ensure the nutrient also, okay, for the coming embryo. This is why we are saying that another difference is there in terms of endosperm. Okay, fine. Remember the two events. Amphimixis is fine. Tropho is related to what? Nutrition. Tropho is related to nutrition because the endosperm is nutritive tissue. That's why I am using the word trophomixis here. Although you are not finding this term commonly in the books, but it's true. It is called as trophomixis or vegetative fertilization, else triple fusion. That thing, fine. So let's compare the endosperm in two different places. Endosperm is nutrient of embryo. Now for sure embryo is fertilization product, means formed after fertilization, no doubt about this. But when we are comparing it in gymnosperms and angiosperms, those cases are going to be different. How? Here it is. In gymnosperms, this endosperm is multicellular. Number 2, haploid as female gametophyte. Okay, and one more thing. And what is that? that after these things, multicellular haploid as female gametophyte, right? All these things are mentioned about the endosperm of gymnosperms. Whereas in the cases of angiosperm, it is mostly multinucleate. The ploidy is 3N. You can write it as triploid. 2N is diploid, 3N is triploid. As formed as female gametophyte. It is not like this. Clear? The fate as gametophyte or sporophyte is not mentioned. Okay? For them means gymnosperm, I can use the word pre-fertilization. Therefore, before embryo as well. Here, post-fertilization. But still before embryo. That's it. Easier. Clear? Easier, I think. So this is how we have compared the endosperm in two places. Fine. On the basis of this, on the basis of these points, these are like mammals and gymnosperms are like birds or aves. Okay. So Bryophyta, amphibian of plant kingdom, ferns, botanical snakes, gymnosperm, birds or aves of plant kingdom, mammals of plant kingdom, angiosperms. Post-fertilization changes. Now there are so many structures like oospore, ovule, polar nuclei, integument, ovary and wall of ovary. Oospore means the zygote, the ploidy is 2N. Ovule is again 2N, megasporangium. Polar nuclei are NN actually, and then integument and the other things are given. Okay. Now they are saying what happened after fertilization, what they are going to form. Oospore will form the embryo, that I can write here what? That this embryo is future sporophyte. This embryo is future sporophyte. Number second, ovule is going to be the seed. Polar nuclei is going to form the endosperm, which is 3N. This is nutrient of the embryo. Integument is going to be the seed coat. Ploidy 2N. I can write it as what? Old sporophyte. Okay, fine. Future sporophyte, old sporophyte. Ovary will form the fruit, wall of ovary will form the skin of the fruit or pericarp. So in this way, if anyone is asking a question about the seed of angiosperms, how many generations are there? 2, and both are sporophytic. Seed of angiosperm, 2 generations. Whereas for gymnosperms we have mentioned 3 generations. Old sporophyte, new sporophyte and female gametophyte. Here it is like old sporophyte, new sporophyte, that's it. Nothing is gametophytic. So this is how we need to remember about the changes happening in angiosperms. Okay. So about the angiosperm here we are going to conclude then. What is alteration of generation? So in total of the group, means in the entire plant kingdom or wherever sexual reproduction is taking place in the plant groups, there are 2 generations as we had discussed in the beginning, gametophyte and sporophyte. Where these 2 generations are found, where we are observing the 2 particular events of sexual reproduction. One is called as syngamy, the other is called as meiosis. Particular, definitely they must occur in sexual reproduction, and when these are occurring, definitely the 2 type of generations, means the haploid and the diploid one, the gametophyte and sporophyte respectively, are going to be here, means getting alternated with each other. Because alteration of generation is character of all sexually reproducing organisms, we are saying the meiosis is actually the reduction division. Means meiosis causes unpairing of chromosomes, whereas zygote is having paired chromosome. So what syngamy did? it paired the chromosome. What meiosis did? It separated the chromosome. So that's why counteracts the effect of fertilization or syngamy. I repeat, syngamy is going to cause pairing of chromosome from different parents, right? It is going to cause pairing of chromosome from different parents. Whereas meiosis is going to do reduction division. Now reduction means unpairing of chromosomes. This is the feature, right? So this is how we used to mention about alteration of generation. Now what type of pattern is exhibited? Okay, or which particular phase is more prominent, that is going to be the basis of naming the life cycle pattern. For example, this gametophytic generation is longer, means of more duration, whereas the sporophyte is confined to zygote only. The sporophyte is confined to unicellular zygote only. So here is the thing, that gametophyte is dominant, number 2 unicellular zygote is the only sporophytic stage, and thirdly exhibit zygotic meiosis. See where the meiosis is taking place? In the zygote. That's why I have mentioned that zygote is the only sporophytic stage, means only diploid stage is zygote. Most of the time, or you can say almost complete of the time, is getting spent as gametophyte, means the haploid. Where we are finding them? We are observing them in most of algae. I repeat, most of algae are showing the same thing. This is called as haplontic or haplobiontic life cycle. In the other cases, see zygotic meiosis, na? The entire life cycle is more or expressed as gametophyte. So many places you will find a one more word about zygotic meiosis. And what is that? That zygotic meiosis is called as initial meiosis also, because the one whose majority of the life is spent as gametophyte, for that one the spore is formed from meiosis. The first stage is formed from meiosis. That's why initial meiosis. Okay. Second type. The second type of life cycle pattern is diplontic. Now, as the name says, diplontic, diploid. Yeah, the diploid is longer one. The sporophyte seems to be multicellular here, and it is going to form the structures by meiosis. What are those structures? Those are maybe forming the spores, resulting into gametophyte. The gametophyte will bear the gametes. See, in the entire plant kingdom, gametophyte only forms the gamete, or you can say the gametophyte is the structure which is what? Haploid. This is seen in most of the seed plants as well. Wait for it. But there are certain examples of algae where we are observing that meiosis happened and gametes are formed directly. So both the stages or both the type of example in some algae and in all seed plants, life cycle pattern is diplontic. Now how come the two are getting different? See the things. The sporophyte dominates. Gamete is the only gametophytic stage for what? Certain algae like diatoms, where we are observing which type of meiosis? Gametic meiosis, because gamete is the cell of gametophytic generation and the sporophyte is undergoing meiosis to form the gametophytic generation directly. Here I can say the gametic meiosis is also called as terminal meiosis. Okay, fine. Remember, I'll remember it for always, that zygotic meiosis is initial meiosis, gametic meiosis is terminal meiosis. To form gametes, terminal meiosis. Okay, in certain algal example like diatoms, there is no prominent gametophyte or there is no distinct gametophyte. Their gamete is the only stage which is haploid. In the seed plant, however, gametophyte is formed but is of shorter duration and is dependent one. So a reduced dependent gametophyte developed through sporic meiosis. Here the meiosis is different. It is sporic meiosis now. Now the sporic meiosis will be like in between gametophyte and sporophyte. One stage came after meiosis, spore developed into gametophyte, then gametophyte bear the gametes, and then the whole sexual reproduction happened to form the sporophyte, which is distinct. So, sporic meiosis is mentioned as intermediate meiosis. Sporic meiosis is intermediate meiosis. So, I have three types of meiosis taking place. Sporic meiosis, intermediate meiosis, gametic meiosis, terminal meiosis, zygotic meiosis, initial meiosis. Okay. So, seed plants are showing diplontic life cycle, but which type of meiosis? Sporic meiosis. Some algae are showing diplontic life cycle, but which type of meiosis? Gametic meiosis. Most of the algae are showing zygotic meiosis. Okay. The third type of life cycle pattern where gametophyte and sporophyte are becoming more and more clear. Okay, where they are both present. It is not about dependent or independent, it depends where it is. Okay, where we are observing. Again, here sporophyte is undergoing sporic meiosis. Okay, wait. In all the land plants from bryophyta up to angiosperms, sporic meiosis is present. I repeat, sporic meiosis is present in all land plants. Okay, but in the cases of bryophyta and teratophyta, these two only are left. We have completed gymnos and angios, means the seed plants. We have completed the most algae also. What is left now? Bryophyta, teratophyta, maybe few algae. So, sporic meiosis is taking place. The spores are germinated into gametophyte, then a longer duration of gametophyte again. I am not discussing anything about dependent sporophyte or independent gametophyte, or both are independent or something like this. I am just mentioning the sporic meiosis happened and a considerably longer stages gametophyte, sporophyte are observed. So, here it is like sporophyte and gametophyte are present as distinct phases, and if they are in distinct phases, the gametophyte is developed through sporic meiosis. So, the sporic meiosis came again. Sporic meiosis can be seen in one, diplontic life cycle, second, haplodiplontic life cycle. Okay. Next, all bryophyta with independent gametophyte and dependent sporophyte are showing haplodiplontic life cycle pattern, whereas all teratophyta with independent gametophyte and independent sporophyte are showing haplodiplontic life cycle pattern. Fine. So, this is fine. This is how we can explain the things, right. So, these are the life cycle patterns that can be seen. The haplodiplontic life cycle can be seen in some algae also. And what are those algae? They may be brown algae like Ectocarpus, Laminaria. They may be some red algae like Polysiphonia. Polysiphonia, previous year question. This is how we need to explain. That's it, easier one. So, life cycle pattern, what type of questions were there in earlier exam? It was like direct. They are asking name and the type of life cycle pattern exhibited. Nothing about ambiguity, nothing about confusion in the theory. Okay, but if you need to make it more complicated type in terms of language, see this. Try to manipulate, try to understand the thing in a better way. See this one. One is called as, the first one was that we studied was zygotic meiosis. Here we are taking the thing in opposite way. So, my first question is where these are taking place. Okay, zygotic meiosis is observed in most algae, where life cycle pattern is haplontic life cycle. Means where zygote acted as meiocyte. Zygote undergoes meiosis. Sporic meiosis is seen in land plants, whether gymnos or angios. Okay, the life cycle pattern are either diplo, haplontic or I can say haplodiplontic, other one is diplontic, both places. In this case, where gametic meiosis is seen, some algae. And the life cycle pattern is again diplontic. Okay, if this is the condition, here I can say one more thing about them. What? That what is the fate of zygote? Means what zygote is going to do. In this particular example of gametic meiosis, we are saying the zygote is going to be what? Gametic meiosis, we are saying to form the gametes. Okay, and in this case we are saying zygote is going to be mitocyte, means undergo mitosis. Sporic meiosis will be, in the cases of sporic meiosis, again we are saying the zygote will act as mitocyte, means undergo mitosis. Whereas in the cases of haplontic life cycle pattern, the zygote is acting as meiocyte, means undergo meiosis. Fine, totally different. This is how the things can be manipulated, means converse. They can ask in the opposite way as well, from here to this side. Why I am comparing in this way? Because there is another chapter named reproduction in organisms, where these things are again asked about the fate of zygote after fertilization. This is about the plant kingdom. Here we are going to conclude the chapter. Hope you are going to get the classes very clearly, or you are getting the subject clearly. So do practice the DPPs, the daily practice papers, so that you can make yourself more efficient and more accurate, because only these things are going to help you in the actual exam time, means the NEET 2022. And perfect, obviously to be more perfect, better to follow the classes regularly. Don't miss the classes. Thank you for today. The next topic is going to be the cell as a basic unit of life. So be prepared with your NCERT for that particular chapter. We are going through the NCERT definitely. We are going to study in depth about the cell structure in the coming classes. Thank you for watching. All the best.